Hello, everyone. Welcome to Biognosis seminar. Thank you for joining us to see how a Spectronaut software can elevate depth and scale of proteomics in DIA acquisition mode. Today we have an exciting program. I will start with a short introduction to our software, Spectronaut, and afterwards we have two guest seminars. First by Ben Collins, Queen's University Belfast. He will talk about applications and experiences, how to run DIA passive with Spectronaut in the Linux high-performance computing environment. And afterwards, we will have another guest speaker, Yansheng Liu from Yale University School of Medicine, talking about advanced multiplexed DIA and using it for quantification of the protein turnover impacted by gene copy number alternations. So let's start with the short introduction. Spectronaut, our flagship software, is dedicated to DIA proteomics and supports such workflows as direct DIA and library-based DIA analysis. It's a part of the bigger software family, including also SpectroDive for targeted proteomics and SpectroMine dedicated to DDA proteomics. And here I would like to have some highlight for SpectroDive, as this week we are also launching new version of SpectroDive, SpectroDive 12. Coming back to Spectronaut, it features such workflows as direct DIA, and this is the best performing library-free workflow for DIA analysis available. It has AI in the core of the development, and it has the broadest support for the PTM analysis. This year we launched extension to this PTM workflow, including normalization by the input sample and stoichiometry calculation. Also, Spectronaut offers unlimited throughput, and this is specifically with this high-performance computing environments, including cloud environment as well. And finally, it is all-in-one solution, allowing you to analyze data from the raw files through visualizations, post-analysis, and allowing extensive reporting. I would like to also share a few highlights from our ASMS launch of Spectronaut, Spectronaut 19. Every year we are working on improving performance of the software, and we are benchmarking this extensively. And here you can see that with Spectronaut 19, benchmarking 130 different data sets, we achieve a much big improvement in the total identifications on the protein group and peptide level. Also, what is very important, we achieve big improvements in the quantification and in big controlled quantitative experimental data sets, 40 different data sets, we observe plus 11% identifications recovery of the differential abundance candidates. And also what's very important for the high throughput, which is the biggest trend now in the proteomics, is we are performing support for the big data sets, and for that we reduce the total RAM needed for the quantification in the big direct DIA experiments, achieving up to 90% reduction of the total RAM required, and 36% reduction in the total analysis time. And with this, I would like to thank you, and I will give a stage now to our first guest speaker, Ben, please.
Okay. Great, thanks very much, Monica. I'm very happy to be here to show a little bit of our work, and we had a very nice interaction with the Biognosis people over the last few years, and send them many emails with questions and so on. So yeah, it's been it's been a nice few years. Yeah, I have basically two topics on the menu here. I think we'll first try to talk about what Monica mentioned in terms of can you run Spectronaut in a high-performance computing environment with Linux and so on. And we've been doing this for about the last year, but in the last 2 weeks we've tried to make some improvements on this. And the second will be kind of more of an application using DIA passive. And the last couple of years we've become quite interested in the kind of drug discovery space and how proteomics can be used to support that. So let's start with the HPC stuff. By way of motivation, actually, I want to say the reason why we want to go in the direction of these kind of higher throughput measurements is because we recently started a center grant with a drug discovery company called Almac Discovery, which, yeah, this laser pointer is not super fantastic. Anyway, where we tried to set up a chemoproteomics center of excellence. And of course, we're trying to bring the proteomics side of this, as well as some PIs from the university bringing kind of physiologically relevant cell models in cancer and innate immunity, and Almac are bringing the medicinal chemistry and kind of high throughput computational biology experience. And so the first sort of 8 or 10 months of this grant has been really trying to set up this first part, this kind of high throughput protein measurements that are going to be compatible with the kinds of screens and so on that our partner would like to do. And eventually, first we're looking at trying to look at targeted protein degradation and eventually in the direction of protein covalent ligandability. So yeah, I guess I want to make the somewhat possibly obvious point that many universities have these high-performance kind of computing centers. Our one is called this Kelvin 2 cluster. It's not especially large, but it has 100 compute nodes, each with 128 cores and RAM and so on, and high memory, GPUs, all the rest. This is actually a national facility that's hosted at our university, but I guess the reason why I put this slide here is to say maybe, at least for the academic people in the audience, possibly you probably have access to such a facility as well, whether you know about it or whether you use it. And there is some kind of barrier to entry in terms of learning how to interact with the cluster job scheduler and scripting and so on. But once you do, then there's very often quite some resources there that you can use, and we decided we wanted to try to take advantage of this. So I think there have been some key developments in Spectronaut over the last few years. Well, some features are older, some features are newer, that are allowing us to try and generate these workflows. The most important one is Linux support via these .NET libraries that came in the last year or a little bit more. So this is crucial because basically, the majority of these HPC centers are working on Linux. Also very useful is the possibility to get a multi-user license. So the idea that you would have like one license key, but are able to run several concurrent instances of Spectronaut on, for example, different compute nodes. This is now a possibility. A floating license, this also is actually quite important. It turns out that the Spectronaut license is activated, deactivated automatically because in the past you had to do this explicitly, and this was kind of problematic running in a kind of parallel way. So this is very nice. Also core, I think, is this saved experiment file, this SNE file that you'll probably be familiar with if you're a Spectronaut user. So this allows you to do the sort of heavy signal processing on the HPC and then move this saved experiment file to like a local Windows machine to allow you to kind of visualize the data and potentially to do some kind of post-processing, some whatever renormalization or threshold changes, these kinds of things. There's also the possibility to merge these SNE files. So if you have multiple DIA analysis, you can kind of merge them together. And this is also, you will see, important for this kind of parallel processing. And yeah, as I mentioned, you can do the visualization offline. Similarly, for spectral libraries, Spectronaut has the spectral archive format, this PSAR file that's generated when you run a library, and we have the possibility to combine these spectral archives, which are basically unfiltered libraries. Essentially, we can combine them into a library, and Spectronaut will handle recalculating the FDR and combine it in an appropriate way. So all these features kind of collectively is allowing us to try and go after these kind of cluster-based workflows. So I want to mention about 3 kind of roughly modes of running Spectronaut that we've been doing. So we started using this, the software about 4 years ago, and we did kind of what most people are doing probably, which is to run on some local workstation with a decent amount of RAM and some multi-threaded processing. So we did that for some time. Then about a year ago, we said when this Linux capability came out, we started to run what we would call like a single node cluster workflow, where we're basically going to the cluster and saying, just give us one compute node, and we will run like kind of in the same way we're running on the Windows one, except from the command line, one Spectronaut job on one compute node. And so, for example, we can request whatever 64 cores and some number of RAM from the cluster, and it will be assigned to us, and then we run the job and so on. But this is not really particularly parallel. It's like doing the same thing as on the Windows box, but on one node. But we have been using this workflow for the last year, and it does give us access to more compute at the right price. So we've been quite happy with this. In the last, I would say now 1 or 2 weeks only, so this is rather new, and I got like the latest data from the postdoc last night. We've been trying to rather take advantage of more parallelization. So running Spectronaut on multiple instances, multiple nodes of the cluster. And so in this case, in the benchmark I will show, we were running a maximum of like 4 compute nodes. And for this, what you need to do is to be able to, chunk the data. So run, some raw files here on this node, some raw files on the other node, and later combine these, both for the library generation and for the DIA analysis. And for this, we're using a workflow management system called Nextflow, which is a bit more flexible and is handling, the job submission to the Slurm job scheduler, and it will be a bit more portable to like different high-performance computing setups, we think, if we use this Nextflow pipeline. So here's a quick benchmark. We didn't have a lot of time to do benchmarking with this new workflow, but this is a very quick one in terms of just how long it takes to run these things. So this is 31 DIA PASEF files from some THP cell lysates. We're running 40 samples per day, and essentially we're trying to do a direct DIA analysis, or at least on these first 2 we're doing a direct DIA analysis, and on the last we're trying to kind of recapitulate a direct DIA analysis, but in sort of chunks. And you can see that the Windows one actually goes pretty fast. It's pretty well optimized. With this 20-core i9 processor. The HPC on the single node, even though it's 64 cores, actually takes a bit longer. These are slower. It's a slower base rate processor, so I think 3.7 gigahertz versus 2, so that explains some of that, but maybe there are also some bottlenecks in terms of file I/O on the cluster that need to be sorted out. We haven't worried about this too much. We just simply ask for more processors, which I realize is not a good philosophy, but you can drive this time down if you want to by just asking for more resources. But recently now, a new postdoc in the lab, Chimaya Narayana, has tried to do this kind of more parallelized way, and it indeed seems to go a bit faster. However, we've just set this up, and we think there's plenty of room for optimization, like asking how many raw files you send at once to one node and so on. And we can also, in a custom way, ask for different resources for different parts of the workflow, which makes it a bit more efficient. So just to give a bit of a deep dive on this Nextflow pipeline that we've worked on in the last couple of weeks. So everything inside this kind of green box is happening on the HPC, on the cluster. Everything inside one of these gray boxes is happening on a single compute node of the cluster. And so essentially what we're doing is we're taking, however many raw files we have, and we're sending them one by one for a pulsar search. And each of these single raw data files is producing one of these search archives. And then we collect up all the search archives, and of course you putting in some FASTA and some parameters. We're collecting up all of these search archives and combining them into a single library and generating this KIT file. And then we go back to the raw data file, and it can be a different set than this raw data file, but if you wanted to kind of recapitulate the direct DIA workflow, it would be the same set. And we use this library now to analyze one by one, again in separate single processes, each of these raw files using this library. And each of these generates a single one of these saved experiment file, this SNE file. And now we're running this merging job, so we're merging all these SNEs together, and all of this post-processing, normalization, and so on statistics is being computed at this stage. And we can then, as usual, take this SNE file and bring it to a local computer to do statistics, or data visualization, or the built-in statistics in Spectronaut, or whatever kind of downstream statistics that you are interested in, like in OR. Okay, so this is the HPC part, and I should say actually that we have analyzed much bigger data sets using this workflow than 31 files. This works for 800 files also, but I just show this benchmark as with this newer workflow we haven't quite worked out all the kinks and got a chance to kind of benchmark it on 800 files. Okay, so let me now switch gears to this DIA PASEF and targeted protein degradation. So I suspect many of you are kind of familiar with this idea of targeted protein degradation, but maybe there are some people who are not. So just to give a very brief introduction, the classical way of drugging a protein with a small molecule is to say, here's my protein, there's a pocket here that's, if it's a kinase, it's an ATP binding pocket, and what we're going to do is find some small molecule that fits nicely into this pocket, and now this kinase is inhibited. But lately, the drug discovery people are becoming very interested in the idea of targeted protein degradation, where you would rather try to do something different. You would use the degradation machinery of the cell to degrade a particular target protein. And the idea is that you have this small molecule which will bind to your protein of interest, but it will also recruit, for example, a ubiquitin ligase complex, which is going to ubiquitinate the protein of interest. It's going to get degraded, and you will have some, hopefully, therapeutic benefit based on the removal of this protein from the cell. For this, for establishing the methodology, we picked a very well characterized, this class of compounds is called PROTAC, which is one of the molecules in the field that everybody uses for positive controls and proof of concept. So we also did that. That was developed in Alessio Ciulli's lab in Dundee. So it's consisting of this JQ1 ligand, which is targeting BRD4 and other bromodomain proteins potentially. And you have on this side a ligand for the ubiquitin ligase VHL. So it's basically what's going to happen is what I said. So the target in this case is BRD4. The ubiquitin ligase is VHL, and hopefully you're going to get degradation. I show this picture because only to say that there's a boatload of literature on this MZ1 compound and kind of compounds derived from that. And so you can see they've done very nice structural studies to say, okay, here's the BRD4 target, here's VHL, this little yellow blob in here is this MZ1 compound, and it's basically you're getting this ternary complex constructed, and you're getting ubiquitination of the BRD4 and then degradation. So I just mentioned this to say this is a very well-studied compound, and that's why we chose it. So we set up an experiment, kind of proof of concept experiment for this, that can we do this in sort of, higher throughput. So this is now a 96 well-plate culture using THP1 AML cells. So this is a suspension culture. We put 150,000 cells per well. We have 6 treatment time points. We have 3 concentrations plus the vehicle and 4 replicates. And so this is really what we're going after. We want to just be able to do things in plates all the time now to kind of boost the throughput. The experimental data acquisition workflow is to make a very straightforward lysis, so using deoxycholate, TSEP-CAA, and we just put it in heat for 5 minutes, 95 degrees. There isn't even actually a spin here, so it's very straightforward. And then in this case, so we used 150,000 cells, but we're taking about a quarter of that, so roughly 40,000 cell equivalents to put onto the robot, and we're using this PAC SP3 digestion more or less as implemented by Jesper Olsen and the Evosep guys. And we like this very much. It takes 1.5 hours to set up and maybe 6.5 hours to run this digestion. And out the other end comes, a 96 well rack of these Evosep tips, and you just walk over and put them on the Evosep. In this case, we're using, I think, 50% of the digest, so around 20,000 cell equivalents. I think we can also go much lower in this. And we're using some DIA passive setup with variable windows, using this whisper zoom method from Evosep. So even though we have a reasonably kind of largish amount of material, we're finding this whisper zoom method is working quite nicely, even up to 50, 100 nanograms of material, for example. And that's the setup. So if you're a Spectronaut user, you'll be very familiar with these views where we say, okay, it looks to be like very nice consistent data where we have 8,500 or 9,000 proteins quantified very consistently. Here's the target of this degrader, BRD4, and you can already just eyeballing it see, we have some degradation. So here's a few selected volcano plots where we're comparing let's see, so 100 nanomolar of the compound divided by DMSO, and already at 30 minutes you can see this BRD4 target is starting to kind of move to the left of this volcano plot and starting to be degraded. Out to 2 hours we see kind of quite clean degradation of BRD4, as well as one of the off targets. In a sense it's an off target, and BRD3 as well. And we have a little bit background there. We can keep going throughout the time course. If we go to 4 hours, we're getting stronger degradation of these compounds. ends, BRD2 comes up. So this is all recapitulating basically what's happening in the literature. If you go all the way out to 24 hours, actually the cells start to go, you get a lot of noise, I would say biological noise, right? So kind of transcriptional downregulation and so on. So usually our target for this would be kind of 4 hours, where the effects might be a little bit more specific. And now that we have, similar to what Bernhard showed on, I guess it was Sunday, if we can do multiple concentrations, we can compute the Dmax, so the amount of protein degraded as a fraction of the total, right? So at 30 minutes, we have 35% of this BRD4 degraded, and it gets all the way out to sort of 80% at 8 hours or 24 hours. We can estimate these DC50 values and do all these kinds of things that the drug discovery people enjoy. Yeah, this is basically the same, but just to show that we can overlay these response curves. We can also look at the kinetics now, right, for a given concentration, right? So in the DMSO, it's flat, but at 10 nanomolar, we start to see degradation already, and we get a different kinetic with kind of higher concentrations. And if you want to be fancy, you can kind of make these contour plots, right? So this is the degradation space for BRD4 along the concentration dimension and the time dimension, and the response, like how much is being degraded, right? So this is the kind of maximum degradation down here. And I think this is common to do in the drug discovery space, but usually they're doing it with looking at these kinds of things with a single protein. But we can have 8,500 of these plots if we so desire. Okay, so I think that's pretty much it from me. I mean, the take-home messages are that for chemical biology and drug discovery applications, we definitely feel like we need to scale up. So that's our kind of motivation. Either you want to be able to do many compounds, or you want to be able to kind of do kinetics or dose responses. We feel like we have now a workflow that's working pretty well for global proteomics in these perturbation screens, relying on like 96 well plate cell culture, relying on this kind of EvoSep, open trons, packed digestion type of workflow, which we're very happy with, and DIA-PASEF, along with this cluster-based version of Spectronaut processing, which we're admittedly still working on, but it's already our main workflow. We're very happy with this cluster-based processing of Spectronaut, and I think there's certainly work to do here. It's far from optimized, and I think this MZ1 degrader data looks just very clean, and we're very happy with this, and we're now at the point where we can start to analyze, let's say, real experimental compounds. So let me acknowledge who did the work here. Mainly this was Mohammed Arefian, who set up the experimental parts. Chinmaya was setting up this NextFlow workflow. Emma was doing the cell culture in this experiment. We also have many other people involved in this center from Queen's, including Bobby Graham working on the proteomics, and Darren McKenzie, a postdoc who's also working to set up all these methods. Our partners at Almac Discovery, at Biognosis, at Brooker, also the HPC team in Queens, and the funders. So with that, I thank you for your attention, and I'll answer any questions.
Thank you, Ben. Our questions will be held at the end. We will have a common Q&A session for both of our speakers. So now I would like to invite Yansheng for the second part.
Thank you, Monica. Thanks for coming. Thank you, everyone. So I would like to first thank Biognosis for this opportunity so that I can present you some of our recent studies using PulseSelect to analyze protein turnover. So I would like to start the presentation by arguing the importance of gene copy number alteration for human diseases. As we know, the individual genes can undergo depletion or duplication, making themselves the drivers of certain diseases. And the increasing CNA range, such as the chromosome segmental deletion or duplication, may cause some genetic disorders, while the whole chromosome level gain or loss could cause monosomy or trisomy, for example, trisomy 21 for human Down syndrome. And cancer is really often characterized by the high-grade aneuploidy. So it's a very interesting biological topic for human diseases. In addition, CNA may also drive drug resistance. So in this very well-established model, we have this ovarian cancer cell line called A2780. So after the long-term cisplatin treatment, this cell line will be transformed to a cisplatin-resistant strain, so-called A2780cis. And if you look at the previous publications about these two cell lines, you could see there is a huge cytogenic differences. This segmental chromosome deletion or duplication could happen in this cis strand. So this suggests that the copy number alteration could drive the drug resistant or at least associate with drug resistant. Could be a consequence or could be a cause, but it would be very interesting for us to learn. And as proteomics scientists, the proteomics could have a very better chance, I think, to figure out what is happening at the proteome level for this gene dosage change. I want to quickly review this paper as I did in my talk yesterday, but I think it's quite relevant. So here, we actually measured the trisomy 21 using proteome and the proteostasis profiling. So this is a particular monozygotic twin pair discordant for trisomy 21. This means that one of the twins bear additional chromosome 21, and then we could really figure out what is happening at the gene expression level. So we measured the mRNA by RNA-seq. Actually, we used the data from the Nature paper published by the collaborator. And then for the protein level, we measured them by source by that time. And you can see that at the protein level, if you look at the chromosome 21 genes in the trisomy 21 cells, or the twin cells, you would find that the protein response is quite diverse. So certain proteins are buffered back to the 1-to-1 ratio if they particularly participate in a protein complex. So then we also measured the protein turnover by the pulse source or pulse select DIA, and here you can see we can ascribe this to the accelerated protein degradation rate for the complex in proteins. So this plot generally gives us an impression that in the aneuploidy cells, there is a protein complex stoichiometry based buffering mechanism using the active protein degradation. So my lab at Yale actually continued quite a lot in this research line to measure the protein degradation using particularly pulse-select and the DIA, because as everybody knows here in this audience, DIA is very reproducible, and for this particular experiment, it has the design of often involving multiple samples in this time course, right? So it's a perfect match. So we actually worked in this area and published a few papers. In some of them, we optimized the DIA acquisition method in the Orbitrap Lumos system. And we also worked with diagnosis and look at the bioinformatics side to see if we could improve the scoring a bit for the detection, for which I will talk about today as well. So, so there are also some biological applications. Some papers are being summarized. I will particularly talking about this paper in preparation. So I just want to make the brief statement or the comment that the DIA field, as we can witness, is now going to the multiplexed manner, right? For example, from the Plex DIA from Nikolai Salvas's lab and also the MDIA from Matthias Mann's group, you can see that this is really a very good trend to make DIA in even higher throughput, as Ben just nicely elaborated. So this can be very interesting, and we are happy that we contributed to the field in the early days by doing this pulse select DIA analysis for protein turnover. So the senior postdoc, Barbara Sovaska in my lab, actually published this paper a few years ago together with Diagnosis, in which we want to introduce this ISW workflow, the inverted spike-in workflow, into the detection of the peptide pairs in this pulse DIA data. So ISW is actually not new for those who are older colleagues knows the history. So in the older days, when the synthesis of the heavy peptide can be quite expensive, people tend to synthesize the light peptide and then spike the light peptide into the heavy labeled human proteome samples. So then the light peptide are used for the identification only, and then the heavy and the light ratios are used for quantification. So we reason that in the pulse-like experiment, especially in the early time points, the light channels are always higher than the heavy channels for most proteins. Therefore, we could directly import the ISW workflow or the scoring in this data set. And indeed, we find we achieved the 30% increase of the heavy to light ratios, especially for the early time points. So this is very nice. We have more IDs. And in addition, on the right, I just want to elaborate further why DIA data can be quite useful for SELEC or pulse-like experiment. Here you can see we compared to the traditional MS1-based profiling for the time point 1 hour, 4 hour, and 11 hours. You can see the heavy to light ratios incorporating during this time course. And if you look at the MS2 level where we have the DIA data, suddenly we have much more data points because we have peptide fragments. Most of them are, of course, Y-ions, but based on this lot of data points, we could really perform this further interference filtering. And at the end, we will have more data points, but also higher accuracy to quantify protein turnover. So I think that's very nice for us to know, and we want to further extend a bit in this research line. So in the recent years, as you guys are probably very well aware of, that there are different latest generations of mass spectrometry from different vendors. For example, this Ultra from Teams Ultra from Bruker, now it's Ultra 2 now, right? So we just want to gauge a bit how this latest instrumentation could be benefiting for the pulse-like DIA data set. So we collaborated a bit with Bruker, and we generated the pulse-like DIA data set and measured that on our 6-year-old arbitrary Lumens instrument using the classic DIA mass that we are quite happy with 2.5 hours for the measurement. In addition, in Ultra, we used the DIA passive method with 3 passives and 20 DIA windows. And you can see this is the result. So basically, in our hand, you can see Ultra only consumes less than 10% of the total peptide digest and only consumes 1/3 of the measurement time. However, in the meanwhile, it achieved 15% more peptide quantifications and almost 20% more protein quantifications. So at the end, we could quantify the turnover for almost 9,500 proteins reproducibly. And even if you DIA people, you zoom into these data points per peak, people normally don't talk too much about this now, but you can see that the Ultra still generates more data points at this throughput. So this suggests that everyone should buy a new machine. So in addition to the machine progress, we all witnessed the improvement of machine learning of different software generations, as Monica mentioned in her introduction. So for example, this ISW, I think we were quite happy with it for a few years. But then this labeled workflow, which was actually already available in the latest versions of Spectronaut, is doing also a great job in this analysis, especially with the latest Spectronaut 19. So basically, the idea is that in this labeled workflow, you can dynamically consider both the heavy and the light channels for the peptide detection. And with the improved machine learning, we just want to gauge the identification performance here. So we used a 2-channel standard experiment by the heavy and the light versions for a cell line digest. So this is a 2-channel standard experiment. And you can see the LBL workflow actually achieved a consistently better identification rate now as compared to the ISW workflow, especially for those samples in which the heavy signals are much more abundant than the light signals, which is of course expected. What is also interesting is that you could look at this relative scoring rate in these dilution curves, those scoring rate that is used to identify the heavy and light transitions per peptide. So very nicely, you could say that this now novel machine learning could decide to use more light in the early time point, and then more heavy in the later time point. In this case, it used more light for those samples who had more light signals, and more heavy for those samples who have more heavy signals. So this is very nice for us to benchmark. And then we also quickly benchmarked a 3-channel select experiment using a nice data set published from Kingsfield group recently. So here they have not only the light and heavy, but also the medium version. And you can see they also generated a standard data set with the 3 channels spiked into 2 mixtures with different concentrations of heavy, medium, and light. And we can see nicely that LBL also performs better than ISW. Especially, you can see that in mixture 2, the light channel only accounts for 20% of the total peptide. Therefore, ISW actually fared quite a lot for the mixture 2. And again, the relative scoring weight, this histogram suggests that the machine learning intelligently decided to use proportional transitions that is consistent to these mixture configurations. So after the identification, the latest version of Spectronaut can also allow this channel-specific FDR filtering, which is quite important because we want the FDR filtering to be transparent, right? So the group Q value is actually quite smart. In this case, this is what I told you. It is a combined scoring for both heavy and light channel, or in 3-channel experiment, then it will be a combined scoring for all the channels. However, now Spectronaut 19 also allows the mini-Q and max-Q filtering. In mini-Q, as you can see here, either channel could be relied if they pass the FDR. And for the max-Q value filtering, both channels or all the channels have to pass the FDR. So we also look at a real data set of this A2780. And you can see from the identification point of view, the group Q and the mixed Q are comparable. And in certain cases, group Q can be even slightly better than the mini Q because it really combines the information from both channels. And the max Q, as expected, generated less identifications in the early time points during the labeling, as expected, because the light channels are much higher. And here you see a deviation of the ratio for the identification, for the quantification, but that's actually is because a small fraction of the peptide are identified. So if you look at the same peptide, they are actually the same. So I don't want to confuse you guys that you need to make choices for this different FDR filtering. So in fact, from our evaluation, we could see that the group Q and mini Q are always quite comparable, and both of them covers 99% or even 100 of the peptide quantities we determined from the max Q value. So, so then for you, the question would be, do you want to first go to max Q to look at those very precisely quantified peptide pairs, and then to do your biological investigation? And maybe at the next step, you just enable group Q or mini Q to look at more data points. Of course, they are noisier, they are of lower abundance, but they are also correct in general with their peptide ratios. So with this established, we actually applied this system recently in this A2780 cell lines, which is a aneuploidy cell line, as you can see. So here you can see the mRNA profiling, the protein profiling, and then the protein degradation rate profiling for these cells, and compared between the cis and the parental cell line. So you can see there are a lot of cytogenic change, right? So on the right, you could see a very interesting plot for which we are quite happy with. So this plot actually depicts the fold change at the mRNA level with the fold change at the protein degradation level. So basically it's a transcriptional change and a post-translational change. So we find that this correlation is actually more informative to infer the buffering, because one is generating, one is degrading. So in this case, you can see, in general, this correlation is mild for the proteome, suggesting that mRNA general perform, the degradation general perform a fine-tuning mechanism to regulate the protein level. But importantly and very strongly, you can see if those genes are participating in a protein complex, according to the quorum annotation, for example, this correlation is very strong. It's more than 0.5. So this strongly suggesting the protein complex-based buffering can be reflected by the protein degradation measurement and the mRNA measurement. So this slide is very simple, for which we just want to elaborate that both protein abundance and the turnover profiling can be useful to understand the drug resistance or cisplatin resistance in this case. So if you use volcano plot and the general statistical cutoff, you can see that this profiling at the protein level and degradation level can be quite complementary. And we are also very happy by this 2D enrichment functional analysis, where you can see this slightly negative correlation between the protein fold change and the degradation fold change between these two cell lines. And we find some very interesting examples of mitochondria-based regulation. So we used the Metascape and generated the network and used the M-code algorithm to look at what kind of network hubs are quite significant in this case. So we were very happy to see this ATP synthesis coupled electron transport subnetwork, because this is highly interacted, and you can see for most of the nodes, you have the key degradation going up and the protein going down, so coordinated regulation. And here you can see an example of this NDUFB11 protein and these ratios of heavy and light across the time. Basically in cis, you have more protein degradation. So finally, we look at the deep map, where you have the gene expression data and all the IC50 data for different drugs, including cisplatin. And we find that according to 154 cell lines, we could identify this association of this particular protein, because the less gene expression is indicated for more cisplatin resistance from the deep map, which is a strong validation. So to summarize, what I try to present to you today is that we started from data from different instruments, and we find Ultra actually is doing a great job in profiling the protein turnover. And we also reviewed this labeled workflow and found that it's quite powerful in deciding the scoring weights to identify the heavy and the light peaks. And this channel-specific FDR will allow flexible options for our data analysis. So I didn't have time to show you that we also want to put in together with this bioarchive paper. Hopefully we will submit over this weekend to include this key dagger software that we normally use to process the data, to do virtualization, to summarize the peptide data from the peptide to the protein level with different options. And we benchmark the 2 channels, 3 channel, and we have this interesting story with cisplatin resistance. So finally, these new features, I think, will be very soonly available in Spectronaut 19 in just a couple of days. And here you really see the scoring weight during this labeling. You can easily imagine this can be also useful for quality control for your SELEC experiment or M-track-based quantification. And here you have this plot, which is a scatter plot along the labeling that it will be also available. So in general, I would say Spectronaut is providing a stronger support in SELEC or Plex DIA experiment or Plex DIA, or multiplex DIA experiment. So with this, I want to thank the lab, especially Barbara Sowaska, who was really leading this development together with Oli and Tejas and Lucas from Biognosis, and of course Booker for this generation of the Ultra data set, and my funding, and thank you to be here. I'm here for questions.